to Big Imaging, Lecture 8, Part 3, Sensitivity Analysis, Reproducibility and Plotting. When you have built your algorithm, you need to check its sensibility to different parameters. And for that, I now have a little workflow where we put in some raw images. We have some parameters. So in this case, it's the width of the Gaussian filter and how many neighbors it's working with. Then we have a threshold, which is set to 100 in this case. And based on the filtered image and the threshold, we do some thickness analysis and shape analysis. The different boxes here show what type of value it is. So the green is the input, the blue is a function, the rectangle is a parameter that is used, and finally we have the output. So we have three parameters that we can control in this example. We have the Gaussian filter with its neighborhood and sigma, and we have threshold level. Now we can do a parameter sweep on, for example, the threshold value, and here we have different samples and see what happens with the volume when we run on different thresholds. And we can see here that in the beginning the volume is greater and then it's decreasing for increasing threshold value, which is in principle makes sense, because usually when you set a more strict threshold, the size of the object decrease. And when we do the analysis, we should vary the parameters within a reasonable bound. That we have to judge qualitatively. Say, in this case, it was used 2000 till about 9000. That made sense for this kind of data. But that you can see even from the histogram that in which area it makes sense. So, and then is the question if it's always the same distribution we get. So in this plot, we have a so-called violin plot, where you can see a kind of a histogram of the values. And for the first value range, the shape is about the same of the violin. And then at 7000, all of a sudden, a lot changes. So that's an indication that here is our threshold level where it doesn't make sense to decrease or increase the threshold anymore. That is harder to see if we look at this kind of scatter plot. In order to do the analysis more quantitative, we need some kind of sensitivity metric. And in control systems, where you have voltages, input-output voltages, it's quite easy to do this, because if you have a change in the input voltage, you get a different output voltage. And then the sensitivity is defined as the absolute delta in metric divided by the delta in the parameter. In image processing, it's not so easy to use that kind of parameter, because the S in the control system case is unitless. It's usually voltage over voltage. And if you look at image processing, you have a threshold as a free parameter, and the unit is intensity or gray level, while the metric we want to look at is a volume, which is in cubic meters or some scale thereof. So the sensitivity unit becomes volume per intensity, which is hard to interpret. So what we can do instead is we do some kind of scan where we look at the sensitivity as the maximum of a metric minus the minimum of the metric divided by the average metric. And that gives us some percentage within the range of the parameters changing. So let's look at some real measurements. So in this case, again, it's the thresholding example that we had before. And you can see that the object count changes from some above 1000 down to something quite less. Actually, it's not scaled here on the bar, on the axis. But anyway, you can see that there is a trend in the object count that actually has a a local minimum. And that would be, I would say, the optimum for this thresholding for the object count. If we look at other metrics, we can look at the anisotropy of the objects, also on the volume. You can see here that the sensitivity for anisotropy and volume increase to a point, actually the same point where the count makes a dip, makes the valley. So they point in different ways, but all indicate that we have an optimum at this threshold at about 5,300 or so. So it's not a contradiction that you have one metric going down. It depends on how it is defined. But the important thing is that all make the same conclusion. Sometimes they don't, and then you have to find a choice that where you weight in which is most important. Another thing which is also very important when you start doing your big analysis is to have a reproducible experiment and analysis. And it's a very broad topic and has very many sub areas. We will touch it a bit today, and I think we will also take a view into this in later lectures. And with the main two things we want is having the reproducibility is for the measurement and for the analysis. And in the measurement, it shouldn't be the result shouldn't be sensitive to how you mount or rotate the sample. It shouldn't be sensitive to noise, and shouldn't be dependent on, for example, exact illumination. That should be somehow normalized in the process. And for the analysis, there shouldn't be any platform dependence, so it shouldn't matter which operating system you're using or which language, ideally, but usually we stay on one programming language when we do the analysis. There shouldn't be any proprietary or in-house algorithms, so everything should be open so people can see what you actually did. The manual part, like clicking, tweaking, copying, etc., should be ideally not at all, but if you need it, then it should be at least minimal. And it also makes sense to have a single script that takes you the whole way from image to result, so you don't have to run several scripts in a given order. So the important thing is doing your analysis in a disciplined manner, and for that you should have a clear recipe. You should have some scripts that can be repeated and reproduce the same results always. Important is there shouldn't be any magic, as I say it here. In principle, there's no special clicking or tweaking a parameter for each sample or something like that. And finally, it should be documented. It's also very important that you can document it, and ideally, if you want to share your things, is to put it on an open repository so everybody can see your code that you used for the analysis. So, what are the advantages of a disciplined workflow? So, first of all, having everything automated means that processing 100 samples is as easy as processing a single sample. Of course, you need to pay some extra effort into making it that way, but it actually pays off when you start having large data sets. And the other thing is that it's actually easier to adapt and reuse your analysis for other work. So if you have one really well working script, then you may only need to modify some parameters to address different types of cells or different type of regions and so on. There is, of course, also the sarcastic view of it as XKCD sees it, that in theory you put some extra effort in writing some code and then automation takes over. And the reality is that you start writing code and you debug, rethink, and so on. Reality is actually somewhere in between those two. It depends on your coding skills and how much you have to invent on the way that you do your analysis. But mostly, if you have described your recipe well, so in principle, going back to the previous slide, having fixed and well-defined steps, then you shouldn't go into this trap of having ongoing development that takes forever. So, how can you do your reproducible analysis? We want to perform the same analysis many times, and to understand how well it works, we need to have a certain workflow. So the notebooks that I'm using here for the lectures and you use for the exercises, they're good for developing and maybe documenting analysis workflow. But for doing the reproducible analysis, you should rather go over to writing scripts or function libraries and macros and having general big scripts that can execute your process. So below you can see an example what such a file could look like, where you have first an import of sys to get the command line argument, and then you have your own code, which is in my analysis, and there in that one you have some analysis script. And when you call this on the command line, you get the file name or folder name or something from the call, and then you run your analysis script and out comes the result. Sometimes you even wrap over a loop and repeat over some folder structure of images. But the important thing is actually that you can reduce it to have this kind of structure. The last part for today is how to present your results. So in principle, bring out your message. And no matter what kind of experiment you do, in the end, you want to present your result. The question is how you present it. And depending on the audience, you have to choose the style and the data, the density of information you have on the slides or on the graphs. So for example, if you have discussions with a colleague or your supervisor, then you can have quite heavy, detailed information in your plots. But already the next step, when you go on a presentation on a conference, then you must reduce your plots radically to just show a minimal statement from your plots. And the same goes if you even go to publication, then you need to refine the data or the plots even further. Same goes, of course, for the web page, because there you don't have any interaction with the audience. And the visualization as such is actually a big problem that you have when you try to analyze the large amounts of data, and in particular if it's heterogeneous data. And the first thing is, just as you have seen many times, tables. It's very hard to see what is actually information, where the information lies. And 3D visualizations may look beautiful. You can make fantastic movies where you go through the sample and showing things, but they are more for presentations and for demonstrations than for the true analysis. And what you need to do is actually boil down to very specific plots that shows and highlights the information you want. And the purpose of the visualization is different, and also the character of the visualization follows with that. So one part is understanding and exploring your data. So that is in the initial part of your experiment, and that's usually for a very small and known audience like you and your colleagues or your supervisor. And everybody you talk to, they have a pretty high degree of understanding of the specific topic that you are talking about. Then you can have more detail and more information. But still, as soon as you put too much, it takes longer to understand and longer to discuss what it is. So maybe split still in several plots. Then to present your results, as I already said, you definitely have a wider and sometimes unknown audience. Could be a reader of a paper, someone listen to your presentations. And at the best, they have a general understanding of the topic you're presenting. And this cartoon here from Knaflick actually shows a nice relation. So if you're doing a presentation live, then it's you and the audience. Then you have quite a lot of control. And the level of detail needed on each plot can be very limited. On the other hand, if you have a written document or an email or a web page, then you have need for detailed description, not too much detail, but detailed description. And it should be clear what every piece of the plot means. And the amount of control you have is of course low because you never see the people, or if you're on the presentation, on a conference, then of course you see the people, but you only get feedback afterwards. Then there are of course a lot of bad graphs, and many graphs, they say, my data is very complicated. And the other statement is, I know how to use this and that toolbox in Python, MATLAB, R, Mathematica, whatever. It can be technically detailed and you have a very lot of decorations and things, but they don't really tell much. Then most programs that are out there, by default, they make relatively poor plots. And you need to work on the plots to make them useful. And the truth is, actually, good visualizations take time to produce. And it actually comes back a little bit to the case that you need to know how to use some kind of toolbox. But you shouldn't over-technify it. And a special thing is also that actually this is not completely untrue, that the quality of charts decreased in the early 90s when people had to make their own graphics, and all of a sudden they were overwhelmed with doing this and no one really had the time to think about it. But then there was a new awareness and maybe the programs also improved, and with that we got better plots. But still, I see bad examples of how data is visualized on presentations by colleagues on conferences, etc. So here are some bad examples, and they are cropped from different presentations or web pages. And in principle, what it says, what the bad thing about them is, this case is so overcrowded of information that you don't see anything. This one, same thing. You have in the beginning, you can see some trends, but in the end, where the interesting part probably comes, you don't see anything. This one is very hard to track at all what is going on. There are too many lines. You never know where they're going or anything. And that one, yeah, well, it's just too small. You don't see the text. And with that, it's useless. Doesn't bring anything. So how to improve? Some key ideas on that. First of all, you have to be sure about what is my message. So that is the first point when you start making your plot. Next step is, how can I clearly communicate my message? And finally, actually, is your plot necessary? Can you write it in words? Can you say it? And Usually it is necessary because you already had a message that you want to tell. And these two publications here are good references on how to work with data presentations. So some simple rules are: never use 3D designed objects for your presentation or for plotting. So this should be some kind of bar chart, but it uses cones instead. The question is, what does this cone actually mean? So it's not really useful and it's confusing. Pie charts can be used, but if they're used the wrong way, they are really hard to interpret. And the important thing about pie charts is that you should only use it to show something as fractions of a full population or something. Then use always white as background for your plots, not light gray. Can be discussed if you can use transparency instead, but then you have to adapt the colors of the plot so that it actually works well with the background. Then using color palettes which are adapted to human visual sensitivity in the right way, and colors and transparency should be used in a smart way. So if you follow all these guidelines, then you can get something that is useful. Now the last part is kind of a, call it, philosophical way of seeing how you can work with the graphics. And here we make the parallel to linguistics, saying that there is a grammar of graphics. And in principle, what is a grammar? It's a set of rules to construct and validating a sentence. So you always have the specific order in different languages, how a word should be put in order to make sense of the sentence. And there is always specific relationships and for the order and how these words work together. That can also be done on graphics. So you can see the graphics, the full graphics as a sentence, and the elements in it can be seen as words. And this concept was introduced in this book, The Grammar of Graphics. And there were also some earlier works who proposed to use some grammar to describe statistical graphics. So, what does it look like? In Excel or MATLAB, in Excel you just mark the range and plot, select plot type and click make, and then you get some plot. In MATLAB you say, or in Python 2, you say plot X and Y data with some colors and shapes. And the idea now is to split this process into some kind of words instead. So let's look at the grammar. We have this nice plot with all the elements in it. And the different parts of it is, first of all, we have some data mapping. We have variable 1 and 2, which are mapped to X and Y. We have some points. So now it's not where the points are, but the shape and color of them. Next is we have axes and coordinates. You can have it with a grid or not, if you like. And finally, you do the last decoration with labels and annotation. So then we have the title of the plot, we have the labels of the data points and the axis labels. And with that, we can build up some information or some structure in how to present the data. So the information you need to provide to annotate your items for the visualization is, first of all, for plots. You have some curve legend telling what each curve represents. You have axis labels telling what information you see on each axis. Finally, you have a title of the figure, and if you use multiple plots in the same figure. And for images, You also need to add color bar to tell how the colors represented in the images, how they are mapped to values in the data. A scale bar is also needed to tell the size of the objects in the image, because otherwise it can be anything, can be any scale. And in particular in physics, people really ask for having a scale bar on every image, because they have really a metric physical correspondence. The colors can be used very differently, and color maps is a research area of its own. And this in a paper by Krameri et al., there was giving a decision tree how to choose your color map, and in the end, going through this, you see that there are different types of color maps that you can use for different purposes. So, first of all, what is my message when you start plotting something? You can just show a lot of points, and it's usually not good to just do the basic plot. As you can see here, you can see that there probably is some kind of trend in this data, which by the way is just random. But you can't really tell much more. So if we would add something like a trend line, so then we can see here that we have, first of all, the points, then we have some fitting, and then we have labels, and then we add on some colored theme, and also the size of the text. Next, then we can start doing something else, maybe extract, plot how the density is of the data. This is a different data set than before, which has more focus towards the middle, as you can see, maybe, or at least get an idea that here is higher density than out here. But it's not so easy in the previous one to really say anything about the data density. Now, by changing to group, sub-histogram, then you can see all of a sudden that we have some frequency count in here, that you can even have a trend line. And with that, it starts getting more clear than what your dots showed before. That can be then in different variations. We can have some ISO lines, if that makes sense. That's more a reduction further, because it's still the histogram, but it's much more reduced. So it depends a little bit on what you want to focus on. In the previous one, we really focused on the distribution itself, not so much maybe the trend line. Here, it's more about the trend line and seeing that the data is distributed around it. And that brings us to the end of this part and today's lecture. First of all, there are very many packages for visualizing data in Python. The one we have used a lot during the lectures is matplotlib. And I recommend, if you want to continue using it, to look for this cookbook. There may be new versions of it, and you can find it on ethlib. And there's a lot of code examples how you can present your data. Plotly and Seaborn are different packages. Depending on your own preference, you can use whatever you want. And ggplot is the one I used to use this grammar style plotting. Which one you use in the end depends a lot of your own taste and your own preferences. And if you want to see an overview of these packages, you can look at this webpage that is linked here. And yeah, that was the end of today's lecture. And today we have talked about uncertainties in the measurements. We have looked at some issues with the statistics, and it is quite a wide field, and it's not obvious how to work with it. And there is even at ETH and Zurich University, free consultation with statisticians. And usually what they tell is that you need more data. But anyway, they can help you to structure your analysis, and then you can go on. They will not do your work, but they can at least tell you how to approach it. And actually I missed here the reproducibility part, but that was also a thing, how you should structure your code and your analysis in a way that other people can work with it. And finally, we looked at how to visualize the data so that it makes sense in presentations and publications. Thank you for this time.